It's 6 p.m., but nobody yet has joined. So we'll start the class anyway. Okay. Okay. Welcome to week 8 of Computational Systems Biology. I hope I'm unmuted. Yeah. So welcome to week 8 of Computational Systems Biology. This course is taught by Professor Karthik Raman of IIT Madras, and I'm the PMR of PA, Shomu Sukhamajumdar of IC Bangalore. So in the last class, we talked about FPA flux balance analysis, how to perform it, when to perform it, certain techniques like flux balance analysis, like MoMA, like Room. We talked about this and how we can apply these to find the optimal flux. So basically, optimal uh, fluxes that is required to maximize or minimize a production. So today we'll be talking about mostly synthetic ethers, what these are, some gene-protein interactions, and how FPA and a more modified version of FPA, which is FPA. We'll talk about that as well. Okay, let's get started. So what we start with a small introduction on gene-protein reactions or gene-protein associations. So you have a set of genes, and so it's as we talked about. So one gene, this is on the right hand side. This is a case where one gene is creating a protein, which is actually involved in a few reactions. So it can be it's a multifunctional. It is what is known as a multifunctional protein. So one gene getting transcribed, then getting translated to form a protein, which protein actually gets involved in a lot of reactions or a lot of other functions. So it's a multifunctional protein. So going from left to right, so on the left hand side you have this is a simple association where you form a protein from a gene and which gets involved in a reaction, and this part will be regulated. So one gene creating one protein which gets involved in a single reaction. But this can also happen to two genes are producing two different proteins, but both of them are actually influencing the reaction as a form of an enzyme or a substrate or any other form. So these are what are known as isozymes. So what are these isozymes? So basically these are similar type of proteins which have similar type of function, but their origin is different. So in this case, protein 1 is getting formed from gene 1, protein 2 is getting formed from gene 2, but they have similar function catalyzing a same reaction. So these are what are known as isozymes. So these are multiple genes encoding distinct proteins carrying out the same function. And then you can have multimeric protein complexes when multiple genes encoding distinct protein subunits come together to form an active enzyme. So what it is happening is the, uh, the, the enzyme being a protein will be made up of certain subunits of the protein. So yeah, so it can happen that one gene is producing one part of the one subunit of the protein, another gene is producing another subunit, and like this, like 3-4 subunits are formed from 3-4 different genes, and all of them actually come together to form the complex protein, which will actually catalyze a particular reaction. So these are what are known as multimeric proteins. So the origin of the protein is divided into the origin of its subunits, which are originating from different genes. Different genes are encoding for the subunits, and the ultimate protein is formed by fusing all these subunits together. And the final one, as I previously mentioned, is a multifunctional protein with basically one gene is actually creating a protein which is involved in multiple functions. So these are the different types of gene protein interactions that can happen. Like broadly saying, there can be obviously more complicated interactions as well. But yeah, these are like very examples of very simple gene protein reaction associations. Sorry. Okay. Now we'll talk we'll extend the gene protein interactions and associations, and we'll try to understand reaction deletions and how this can happen. So, metabolic flux balance analysis, which is the FBA, is a computational method that uses genome sequencing and biochemical databases to understand and study an organism's molecular capability, sorry, metabolic capabilities. And FPA can be used to simulate the effects of in silico gene deletions as well. So what we are trying to do is we are deleting a particular gene inside the genome, and we are using FPA to kind of analyze what kind of implications that can that particular deletion have on the total biomass production of the cell per se. The relationship between genes, proteins, and metabolic reactions is often not one-to-one. As I was saying, it's not always the left-hand side case. All the cases are equally probable. And enzymes catalyze reactions, but multiple enzymes can catalyze the same reaction, and one enzyme can catalyze multiple reactions, as the examples that we were talking about. Yeah. Last class we talked about FPA, and this class we are going to extend on it a little bit and try to talk about what can happen when there are certain gene or reaction deletions and how we can use FPA to model this and find out what is going to be the change in the biomass in the whole system. Right. So yeah, so this enzyme catalyze, it's the associations are not always one to one, as I was talking about. So you can have a one to one association like this, but you can also have multiple genes coding for multiple proteins which are involved in a single reaction, or this kind of a situation where your individual protein subunits are actually synthesized by different genes, and all these subunits come together to form the final protein. So yeah, the relationship between genes, proteins, and metabolic reactions is often not one-to-one. Enzymes catalyze reactions, but multiple enzymes can catalyze the same reaction, and one enzyme can catalyze multiple reactions, as I was talking about. Deleting a reaction may require deleting multiple genes, which can alter the metabolic solution space, which is true because like there will be multiple proteins and multiple enzymes. Even if like multiple enzymes are not involved, a single enzyme might be regulated by multiple genes. So deleting a whole reaction is not the same as deleting a gene, because for deleting a whole reaction, you have to delete all the elements that are part of the reaction and all the origins of those elements, of those proteins, etc. So, yeah, and deleting a reaction might actually require deleting multiple genes, not a single gene, which can alter the metabolic solution space. Say, for example, removing a specific reaction may improve production rates, but deleting genes associated with that reaction can actually lead to a synthetic lethal phenotype, as in basically other gene reactions can be deleted. So what you're trying to do is you're trying to delete one single reaction from the metabolic space, but what is happening is when you're trying to delete that reaction, deleting the genes that are associated with this reaction actually is making you a synthetic lethal. It's basically the cell cannot survive, right? So it's not just you identify these particular genes that are involved in making these proteins, and so if you don't want to have these proteins, you just delete off the genes. It's not as straightforward because it might give you a synthetic lethal framework. So, uh, um, and also if the reaction lacks known associated genes, a genetic engineering framework is also not feasible. So actually, this is an example of a gene deletion in a metabolic system. So this is the metabolic model, and this is represented as a combination of three model components. gene 1 to gene 5. So these are the three components. So these are Boolean operators, odd operators associated one with G1, G2, and G3, odd operator also associated with G3 and G4, and a single operator, kind of a direct operator with G5. Yeah, so GPR Boolean rules associated genes, G1 to G5. So these are your five genes and the three reactions. This is first, this is second, and this is third. Uh, and the network of reactions define the set of feasible chemical transformations. And these are basically your feasible chemical transformations. And the set of essential biomass precursors defines the requirement for growth. And the growth phenotypes are predicted by assessing whether all biomass precursors can be produced by the metabolic network from a set of metabolites from the medium. Gene deletions potentially inactivate reactions, which may in turn reduce the space of producible metabolites. So if you, for example, delete gene 5, G5, so it actually deletes the third reaction, and uh, the possible metabolite space is actually reduced from here to here, and one essential biomass precursor is no more producible. So this actually is a synthetic lethal, basically because it's an essential biomass precursor that you're not being able to produce due to deletion of the gene, so the cell might not survive. And in case where a biomass precursor is no more producible, gene deletion is predicted lethal on a given medium. So need to be careful about this. But the idea is this: like you identify these reactions and the genes involved with these reactions, and you try to somehow delete or inactivate those. But then you have to keep in mind of like if it is becoming a synthetic lethal or not. So if actually by deleting the gene, you are actually hampering the cell's ability to survive. So that should not happen. Okay, so yeah, so this slide actually I have put in for so we are talking about deletions. We are talking about deletions at two regions. So one is the gene deletion and one is the reaction deletion. And how actually these are different. So it's not a gene deletion is not equivalent to a reaction deletion and vice versa. So yeah, so this is one example that is given here. So this is a GPR association for trans ketolase reactions. So these are the two trans ketolase TKT1 and TKT2 in E. coli. So these are your genes that are present there, and the mRNA is formed from these two genes. And actually, the gene actually uh, codes for these two enzymes, so TKTA and TKTB. Now, these actually are interconnected in terms of like they regulate each other to form this particular transketolase reactions, TKT1 and TKT2. Now, if you delete any one of them, so basically in this case, you are deleting uh, yeah, so basically, if you are deleting one of these particular reactions, so what happens is, so this is your, so if you do a FBA on this, on the analysis, so you'll get that if you delete any one of these reactions, this is basically your wild type solution space, the blue one, and the green one is your mutant solution space. So your solution will be confined to this particular region. Uh, yeah. So uh, this reaction is actually involved in ethanol production. So this ethanol production is coupled to cellular growth at an improved rate in the mutant constraint. So this is the ethanol production rate, this is the growth rate, and you're trying to optimize using FBA. And this actually increases your ethanol production by deleting this particular reaction. Now, when the corresponding genes actually TKTA, so yeah, so this particular reaction has been deleted in the first case. Now, what you're doing is instead of deleting the reaction, what you're doing is deleting the genes TKTA, TKT, TKTB, PTA, and EUTD are deleted, and to eliminate TKT1 and PTAR, no growth is predicted for the mutant strain as indicated by the solution space only residing in the x-axis. So basically, this tells you that if you delete this particular genes involved in this particular reaction, what will happen is you do not have any solution space. But instead, if you delete one reaction, just the reaction from this, not the individual genes itself, you have a better ethanol production rate. But if you delete the genes itself, you lose your solution space, and basically it's going to be a synthetic lethal, and the strains of E. coli are not going to live. That is it. So now we come to the concept of dead end metabolites. So, okay, so what is a dead end metabolite? A dead end metabolite is defined as a metabolite that is produced by known metabolic reactions of an organism but has no reactions consuming it, or that it is consumed by metabolic reactions of an organism and has no reactions producing it. In both cases has no identified transporter. So the idea is that you know that this particular metabolite is involved in this particular reaction, but you have no idea from where is that metabolite getting produced, or where is that metabolite getting used up. So these are what are known as dead end metabolites, because these are like static in the system, because once there you do not know how it gets degraded, or if it is not there you do not know how it is getting produced. So it becomes a tricky situation. And yeah, so these are actually interesting to study. So these dead end metabolites are thus isolated compounds within a metabolic network. As I said, these are at standstill in terms of concentration, not in terms of concentration, but in terms of like being or not being in the system, right? In some cases, the dead-end metabolites reflect a deficit or an error in how a metabolic database represents knowledge from the scientific literature and alerts us to the need of further curation of the database. In other case, the systems level analysis alerts us to areas where more experimental research is required, because you do not know. Technically, you do not, the idea is you do not know from where the compound is getting generated or how it is getting used up. So, uh, it's necessary to understand in terms of experimentation how actually is the dynamics for this particular compound or the enzyme is. So yeah, so in other cases, the systems level analysis alerts us to areas where more experimental research is required. In the latter case, the dead-end metabolites act as signposts to the unknown of unknowns of metabolism. So basically, you know that the compound is involved, but you do not know about the nature of, not about the production and degradation of the compound. So that is the definition of dead-end metabolite. And this is a representation of generic dead-end metabolites within a metabolic network. So compounds A and C, this is A and C. This is like basically they are not produced inside the system, and you do not know from where they are produced, and but they're involved in the reactions. Uh, yeah, so the compounds labeled A and C are neither produced nor transported by any other reaction within the networks, while compound B and D are neither consumed or transported by any other reaction network. So basically, A and C, you do not know where their origin is, and D and B, you do not know how they are going to be used up in the reaction. So these are these two types of dead end metabolites that we are talking about. Okay. So now we'll talk a little bit more about dead end metabolites classifications. So there are basically two classifications: root non-produced or root non-consumed, as I was discussing in the previous slide and the figure. So there comes the concept of blocked reactions. So initial metabolic network reconstruction often contain inconsistencies due to annotation errors. Okay, yeah. So we have yeah, we have talked about dead-end metabolites. Now we talk about two specific types of dead-end metabolites. One is root non-produced and the other is root non-consumed, as I was talking about in the previous diagram. So in this case, Your C is root non-produced, A is also root non-produced, B and D are root non-consumed. So what it means is basically you know that A and C, all four A, B, C, D are actually involved in the reactions inside the inside the metabolic space, but you do not know from where A and C are getting produced, and you do not know how B and D are getting degraded or used up in the reactions, right? So also we'll talk about a concept of block reaction. So initial metabolic network reconstruction often contain inconsistencies due to annotation errors or unknown enzyme functions. These inconsistencies lead to missing reactions in the draft model, creating gaps in metabolic pathways. So it's basically it's a knowledge gap. So we do not know how these two particular ends are getting connected. We do not know what is the connection between these two particular metabolites or compounds inside the network, right? Yeah, so these are your dead-end metabolites. So the dead-end metabolites are classified as root non-produced or root non-consumed, uh, as I was describing. Root non-produced metabolites, that is metabolites that are not only consumed by the system reactions, and non-consumed are those that includes those metabolites that are only produced by the network but are never consumed. So these reactions, these block reactions, cause further gaps downstream or upstream metabolites. Identifying these inconsistencies is known as the gap finding problem. So it's very intuitive to what it means. It's basically you do not, the dead end metabolites are our ways of telling or understanding that we do not have enough information on the particular metabolite on how it is getting produced or consumed. So, these are what leads to blocked reactions because you do not know. So there will be inconsistencies of these reactions because you will not know the origin and the transportation of these particular metabolites. So, yeah. Now, so how, what are these gap metabolites and how do they look in a pathway? So this is basically a schematic example of these particular dead end metabolites. So yeah, so it's basically describing four classes of gap metabolites, which are shown to as a consequence of missing reactions. So these red crosses indicate the absence of some reactions on the left and the right, and the dotted and continuous arrows represent blocked and non-blocked reactions. So this so the the dotted arrows. So this is a dotted arrow. This is a dotted arrow. These dotted arrows are known as are basically denoting blocked reactions, and the continuous arrows, for example, this and this, are denoting non-blocked reactions. Yellow and green circles represent gap and non-gap metabolites, respectively, and metabolites are labeled according to its class. So in the first reaction, in the absence of a reaction, so if this reaction is not present, uh, in the absence of reaction causes metabolite A to become a root non-produced metabolite. So if you do not have this reaction, this actually becomes a dead-end metabolite. You do not know from where this particular metabolite comes from, right? So this becomes an RNP, root non-produced metabolite, and this affects the propagation. This effect propagates downstream, generating a new gap metabolites, which are basically downstream non-produced and block reaction. So basically, if you do not know where this comes from, this is also a dead end, sorry, downstream non-produced because you know this is coming from this, but you do not know from where A is coming. So B and C become downstream non-produced gap metabolites, right? But here again in D, you have another production pathway, V4, which is actually making this D. So this becomes a non-gap metabolite again. So rest of them, all these three are becoming gap metabolites. Right. So yeah, similar is an example. So in the absence of reactions H, reactions consuming H. So in this case, production was blocked off. In this case, reaction consuming this particular compound is blocked off. And so what it becomes is, so you have E here, but you do not for the upstream for the upstream of A, G and F. You do not know how these are getting consumed. right? So in the absence of reactions, consuming it makes it a root non-consumed metabolite, and this effect propagates upstream, causing other metabolites to become upstream non-consumed in a symmetric manner with respect to the first case. So the idea here is, if you have one dead-end metabolite or one gap reaction, it can actually propagate to the rest of the compounds that are present inside the network, because one gap reaction meaning the rest of the reactions that depend on this particular metabolite will also be somehow affected by the gap. So either it will become a downstream non-produced, or it will become an upstream non-consumed. So yeah. 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 So now we've talked about dead-end metabolites. We've talked about reactions uh, that are actually uh, gap reactions for which you do not know the metabolite, where it is getting produced or how it is getting consumed. So uh, we will actually look into a technique which is very similar to the flux balance analysis that we did last time. So this is what is known as a flux variability analysis. So how is it different? So flux balance analysis predicts steady state fluxes of reactions in a metabolic network based on biological imperatives, which is known. But FPA does not always yield a unique solution. So what it gives you is basically a space of solutions, and sometimes that space is very big, right? Prompting the need for this particular technique. And what it does, it quantifies the feasible ranges of reaction fluxes at optimal or suboptimal production rates determined by FPA. So it's basically an extension of the FPA technique, where you where instead of having a vast solution space for your reactions, in this case, it helps in giving you feasible ranges of reaction fluxes. So it kind of narrows down the solution further, right? So, and this is actually a generalization of FPA, and it aids in understanding metabolic network flexibility. Applications include everywhere. I'll not talk about this, but we'll have a few examples later. It also enables the analysis of reaction flexibility within metabolic networks, offering insights into systems behavior. So what happens is, first, Firstly, you have to identify the maximum value of the objective function and constrain the objective function to this value. Then you minimize and maximize each flux independently to identify the flexibility in fluxes in the alternate across the alternate optima. Right. So this is your original set of solutions from here, and these are your alternate optima. So this is a solution space. This is the alternate optima, same as this, and you're trying to minimize and uh, maximize each flux independently to identify flexibility in the fluxes across the optimal across the alternate optima, right? So if you have n fluxes, it's the calculation goes to around like two n FPA problems for flux variability analysis. Okay, so this is an example. So here you're trying to determine what is possible based on external measurements and network stoichiometry. So you have I metabolites and J reactions here. So this is your feasible region solution space, minus infinity to plus infinity, but you're constraining it into max Vj to minimum Vj, right? So yeah, this is a feasible reaction. Your objective is to maximize and minimize Vj for all I, subject to be in the space of optimal solution. So your steady state condition, which is sigma is equal to 0, you use that to solve the steady state analysis, but you constrain this to V minimum and V maximum, and and yeah, and you have to solve basically two linear optimizations for each reaction J. So it becomes twice the number of problems that you will be solving for the FPA. It becomes twice. Yeah, so the requirement for this particular technique is kind of understandable, because FPA will give you a very FPA will give you a very generalized solution of the metabolic space, but it won't give you a unique solution. So alternate pathways can achieve the same objective, resulting in phenotypically different solutions. So basically, there will be many pathways, many reactions that can actually, different types of reactions that can actually kind of uh, lead to the same FPA optimization result. But these will be phenotypically different solutions. So, uh, although like it's the same, it's the optimal solution space, but you're using different reactions to describe that. So it becomes phenotypically different, right? So what flux variability analysis does is it identifies the maximum and the minimum fluxes through the reactions while maintaining optimal objective values. So flux variability analysis is done using the Cobra toolbox. Will not go into the details of the toolbox. You can look it up in the lab sessions and the tutorial sessions that is there. So you have this set of known functions. This is first one is change reaction bounds. Second is change of objective. Third is optimized CD model. So you have this already given models inside the package. You have to find an optimal objective value for the function, and then you set upper and lower bounds of this objective reaction to this particular value, and obviously set your reaction of interest as your objective, and then you use flux balance analysis to minimize and maximize the new objective separately. This is the maximum and the minimum possible fluxes through the reaction while maintaining optimal objective value. And this is an example of flux maps for two alternate solutions for maximum aerobic growth and succinate. So basically, these are, I think, Krebs cycles. So yes. Yeah, so these are flux maps for two alternate solutions for maximum growth rate on succinate. So succinate is formed during, if I'm not wrong, during Krebs cycles. So these are Krebs cycles only examples. Uh, in the first example, the reaction Me1 is used to convert. I'm not sure if you can see it here. I don't think it's visible from here, but anyway, so there's one reaction called Me1, which is used to convert the L-malate to pyruvate. But in the second reaction, this reaction in the second set, the reaction is not used at all, and the reaction PYK is used. And the two alternative solutions are actually highlighted in red. So this is one alternative solution, and this is the other alternative solution. Right. So these are the like importance and why we should use flux variability analysis in some cases over flux balance analysis. Yeah. So this actually, yeah. So this actually is a basically the code and the methodology by which the previous example was gotten. So you have a Malik reaction, Me1 E. coli growth on succinate. Minimum flux through you have considered as 0, and the maximum you have considered as 6.49. So basically this is the z that you are setting, right? So minimum z is 0, maximum z is 6.49. So yeah, for alternate optimal solutions, one case the reaction is used, and the next case the reaction is not used, right? Now you use Cobra toolbox and try to find which is the better and which is the better solution for this particular problem. So it includes these functions, as I was talking about the flux variability function. So um, this is your actually the code. So basically, first is your model change for reaction bounds, then your FBA solution, then again the models changing objectives and so getting the maximum and the minimum solution. So this brings you to this particular solution. Uh, so these are your minimum and maximum flux given by flux variability analysis. And if you see, so variable reactions for growth on succinate, the uptake rate is 20 millimole and 20 units under aerobic conditions. The minimum and maximum possible flux for every reaction was calculated at the growth rate, and only reactions with variable fluxes are shown here. So these two enzymes, FRD7, which is basically fumarate reductase, and SUCDI, which is the succinate dehydrogenase, always seem to have highly variable fluxes in this model because they form a cycle that can carry any flux. So in a cycle, it's like where you start, you end up there. So it can carry variable fluxes. Yeah, physiologically, these fluxes are not relevant, but the other variable reactions are MDH, which is malate dehydrogenase, Me1, which is malic acid, which is the NAD, Me2, which is the malic enzyme, which is NADP, and then you have other enzymes involved as well. So these are your optimizable metabolites, which you can optimize to get the maximum possible reaction for succinate, growth on succinate. So this is an example on how uh, X variability analysis can be used to to get solutions like this. Okay. So next, what we'll talk about are overexpression algorithms. Just give me a second. Sorry for that. Okay. So yeah, now we'll talk about overexpression algorithms. So basically, here you are trying to overexpress a particular metabolite or enzyme, basically, right? So this is basically uh, your example. So I'll talk about it uh, in the later part. Okay. So the initial fluxes were calculated by constraint-based flux analysis uh, using objective function and maximizing biomass formation. Right. So this is your biomass that you're trying to maximize, VU biomass. Subject to this is your basically yeah, sigma SV equal to 0, which is basically your steady state condition, and where your Sij is your stoichiometric coefficient of the metabolite I in the Jth reaction, and Vj initial is the initial calculated flux of the J reaction. Now, here V biomass, V ATPM, and V ZLYC represent the biomass formation rate, the ATP consumption for rate for maintenance, and the lycopene production rate respectively. And you are trying to maximize this lycopene production, right? So V ATP maintenance is in this case is basically the ATP flux. So these are the things that you're considering for this particular model, right? So you're considering biomass for basically lycopene. Then you're considering the ATPs required for maintenance. Then you are uh, considering the biomass production rate for lycopene, and also uh, the ATP required for non-growth associated maintenance as well. So for initial production rate, you measure it. It's basically measured from the initial experiment, which was around like 0.002 units. It was measured cultivating recombinant E. coli harboring this particular P LYC184 and PTRCDX in a 2 cross YT medium at 30 degrees Celsius. So it was noted that in general, this can be provided as a measure of constraint or calculated constraint if no experimental result is available. So this is the basically the biomass production rate given here. So in this case, this is the algorithm FSCOF. It's a framework to select the gene amplification targets for enhanced product formation. FSCOF searches for candidate fluxes to be amplified through scanning for those fluxes that increase with enforced objective product formation. Enforced objective flux under the objective function for maximizing the biomass product formation flux. During FSCOF implementation, three types of intracellular flux profiles are typically identified: increased, decreased, and changed. Oscillating flux profiles can be found in rare cases as well. And so this is basically from here you are trying to go here. So these are the steps. So this is increasing flux, you're moving up, and this is decreasing flux if you're moving down. Okay, so this is the over expression algorithm. So what you're trying to do again, so you're trying to search for candidate fluxes to be identified through scanning and under the objective function for maximizing the biomass production, right? So you're trying to find a particular candidate when amplified actually maximum amplifies the product formation. This is your uh, objective. Now, how do you implement this overexpression algorithm, right? How do you implement this overexpression algorithm? So these are the components of the algorithm, right? So imposing a taxidine production flux, solving a FPA problem, imposing an amplification in individual reaction fluxes to simulate the effect of gene overexpression, and solving a MoMA problem instead of FPA. We discussed about MoMA in the last class as well. And then identifying overexpressions that led to phenotype fraction value, FPH. Okay. And then 3 and 4 are basically iterated for every reaction within the network. Calculations were made under conditions to simulate complex medium. Medium composition can be approximated by setting uptake rates of specific chemical components known to exist in the medium of interest. The computational complex medium contained all 20 naturally amino acids. Basically, you're assuming that all the amino acids are available for this particular production. And yeah, and there were some lower bounds of amino acid transport reactions set, and they were chosen upon previous literature values because they are satisfying the relative biomass differences experimentally observed in the media, right? So this is the paper actually. If anybody is interested, they can go back and like, check the paper. They actually do this particular overexpression algorithm. They describe this overexpression algorithm in the paper as well. If you want to go and have a look, so that is well. So yeah, so the idea is that you have a base model, you add heterologous components to it. So basically, in this case, yeah, you amplify a particular compound inside the module, and then you calculate an FPA, and then you amplify the reaction, recalculate the fluxes with MoMA, and then identify overproducing strains manually. So this is the overall idea. So yeah, FPA and other techniques, these kind of techniques can be used to over amplify a particular. So the idea is to amplify a particular metabolite, which will amplify the product. So that is the whole idea. Right. Now we come to the final concept of synthetic lethals. So yeah, so we have talked about synthetic lethals a little bit earlier. So what are synthetic lethals is basically it's a concept proposed first in 1922 by an American geneticist called Calvin Bridges, and is observed when simultaneous inhibitions of two genes result in cellular lethality. So basically, two genes that you are simultaneously either knocking down or doing something with it is bringing about cellular lethality that the cell cannot survive. Yeah. Why the use of the word synthetic? The meaning of synthetic used here conforms to the ancient Greek meaning, the combination of entities to form something new. That is what it is here. So classical synthetic lethals occurred when inhibitions of two genes is lethal, while inhibition of either gene alone is not. So remembering this concept is important. So if you have two genes, individually these genes are not lethal. So if you block individually one of these genes, the cell will survive, maybe with some differences in phenotype, but the cell will survive. But when knocked down or inhibited together, the cell cannot survive at all. So it brings about lethality. So that is what a classical synthetic lethal is called. So it occurs when inhibition of two genes is lethal, while inhibition of either gene alone is not. It can be harnessed to treat cancer selectively by identifying interactive genes and targeting their synthetic lethal partner. So there is a massive scope here. So basically, you are trying to target synthetic lethals and basically their pairs, synthetic lethal pairs. So, for example, in a cancer cell, if you have if you know the synthetic lethal pair, so what you can do is target these two particular genes only, and the cell will die. But individually targeting these genes will not work, right? So, yeah, so it can be harnessed to treat cancer selectively by identifying inactive genes and targeting their synthetic lethal partners. In accordance to this concept, synthetic lethals can also be applied to genes and small molecules and can be used to elucidate the mode of action of drugs. Cancer is a disease, it's a multifactorial disease, right? So it's driven by consequences of mutation. So there will be multiple mutations in multiple genes that leads up to the disease. And amplification of genes that allow cells to escape mechanisms that control proliferation and survival. So basically, you'll see an overexpression in genes of cell cycle, DNA repair, and not DNA repair, maybe like not overexpression, maybe underexpression of these genes. So you'll have these, yeah, you'll have these genes that actually control self-proliferation and survival. Hence, inhibitors that target synthetic lethal partners of mutated or amplified genes in cancer cells can cause cancer cell death without affecting the survival of normal cells. So this is very important. So if you can identify these synthetic lethal partners of mutated or amplified genes in cancer cells, you can target these cells and cause cancer cell death without actually affecting the survival of normal cells. So this is actually very, very good to do. This is actually an example of synthetic lethals, usage of synthetic lethals and principles in cancer types. So in this case, a thyroid cancer is shown, uh, uh, and basically in a normal cell, you have these normal copies of gene A and gene B, and in a mutated condition, either gene B is mutated. So yeah. Loss or inhibition of either of the protein products A or B alone, or overexpression of A is viable. However, pharmacological intervention by the partner gene product will result in a synthetic lethal interaction in the tumor cell with a loss of function mutation in a tumor suppressor gene. In addition, pharmacological intervention by partner gene products will result in synthetic dosal lethality interaction in tumor cells with a gain of function mutation or overexpression of the oncogene. The yellow star actually denotes the mutation. So yeah, so basically, if you overexpress, so if basically you are overexpressing uh, uh, by a pharmacological intervention by the partner gene product, and it will lead to synthetic dosage lethality as well. So this technique is very useful for treating disease like cancer, but then again, it's very experimental and needs to be studied a lot. Now, how do you identify this particular synthetic lethal targets? Sorry, yeah, yeah. So how do you identify this synthetic lethal target? So firstly, hypothesis-driven approaches for identifying synthetic lethals. Interactions leverage are known alterations in molecular pathways in cancer, targeting specific genes or pathways based on established knowledge. And also screening, another method that can be used is screening-based approaches involving comparing drug or RNA interference or knockdown treatments in matched cell lines with and without specific gene alterations, identifying or allowing for high throughput identification of synthetic lethal interactions either through chemical library screens or genome-wide interference screens. So basically, what you're doing is in a cancer cell, uh, you have a lost activity or over expressing that particular gene. It's a gain of function in some cases. So basically, yeah. So there are multiple approaches to generate matched cell lines of a loss of function or a gain of function of a specified gene is studied for studying synthetic lethal interactions in cancer cells that have lost expression of a gene. So the cDNA of a gene can be re-expressed in the deficient cell line. So if you take this cDNA and overexpress in a cell line, multiple cancer cell lines, both expressing deficient in the gene, can be investigated. So one is a knocked out version, one is like a wild type version. Then you are investigating the differences. Cancer cell lines that express the gene can be treated to inactivate the gene for studying synthetic lethal interactions in cancer cells with oncogenic mutations that increase the activity of the gene or create a new gene fusion like the BCR-ABL. Multiple approaches can be used. So yeah, here you can identify like first set of figures. ABC is basically telling you how you can identify synthetic lethal pairs and targets using loss of function variations. And the second set of figures, the E and F, are telling you how you can use it using how you can identify using gain of functions. Right? So cells can be treated with RNA interference or chemical inhibitor in order to reduce the expression back or below normal levels. So you have a gain of function, then you are decreasing it to a normal level, and then multiple cancer cell lines, both with and without the mutation, could be investigated, or the activating mutation can be introduced, or the gene can be overexpressed within a cancer cell line that has normal expression of the wild type gene. Now, traditional studies focus on perturbing or deleting single genes or gene pairs, potentially missing out higher order interactions and redundancies in metabolic networks. So this we talked about in the very first class of, of this session. So basically, the idea is that even if you delete one gene, there will be genes that actually are then higher order genes which are actually involved in these functions as well. So it does not actually, so there are a lot of redundancies in the network. So identifying these lethals or just perturbing or deleting single genes or gene pairs actually misses out higher order interactions and redundancies in the network. Right. So extending lethality analysis to triples, quadruples, etc., captures multi-gene reaction interdependencies, but faces combinatorial complexity challenges because the moment you increase the number, the combinations will increase also exponentially. So testing all those becomes actually very difficult. So in this paper, in 2006, they introduced in silico multiple knockout investigation in yeast, cataloging gene sets providing mutual functional backup up to 8 interacting genes. Then again, the same author in 2008 developed a computational approach using game theory for multiple knockout analysis in SCIVC. In 2008, there was another paper which introduced a framework on elementary flux modes for analyzing structural robustness in metabolic networks applied to E. coli and human hepatocytes. Yeast, with its short non-coding regions and genome containing less than 7% introns, is preferred for genetic interaction analysis, with most research focusing on S. cerevisiae E. coli, and E. coli studies are limited due to pairwise interaction. So this is a five-step workflow for the model-guided identification of compounds that exhibit synthetic lethal protein pairs, basically. So first, you build a metabolic model for multiple pathogens, compute synthetic lethal pairs for each model, and overlapping synthetic lethal pairs shared among all models. Right. Then you construct and test double mutants to validate the simulated data. Right. Then you perform virtual screening against validated pairs, and then you test the compound as a single agent in whole assay. And then you check in different combinations. So which of them will give you the best phenotype for your condition should be your ideal synthetic lethal pair. Okay, so yeah, that is all from my side in this lecture. Now we'll discuss certain assignment problems from previous years. So I'll keep the questions on the screen for 2 minutes and then I will discuss. Okay. So this reaction is considering N1 and N2, giving N3 and N4 from gene A and B, right? And you're asked which of the following statements is true if R1 is essential for the survival of the organism, right? So if gene A and gene B, so this is a Boolean operator, right? And is a Boolean operator, so what it means is it requires both gene A and gene B for it to be, for this reaction to hold. So the answer would be option C, both gene A and gene B are essential because there's this AND operator. If it would have been an OR operator, you can say either of them would do. But if since this is an AND operator, then it's like you require both of them together to for this reaction to uh, survive for this reaction to happen. And if this reaction is actually essential for the survival of the organisms, then you need to have both gene A and gene B. So both of them are essential. Okay, so this question is telling you, so you have this reaction N1 plus N2 giving you N3 plus N4, and then the condition is gene A or B and gene C, and then you are asked which of them are essential. So the answer is gene C is essential, first of all, because there is an and either of the pairs, A or C, and C or BC should be functional. So how is it coming? So it's gene A or gene B. Alongside that, you need gene C. See, A or B and C. Right? So AC and BC, that they should be fine. And then gene C is also essential because there's an AND operator, a Boolean operator here. So the gene C is also essential. So two options, third and fourth. So basically using OR, you can either use AC or BC, and also C is important. C should be there. Okay. Moving on. Okay, so the question is the deletion of isozymes of an essential reaction will kill the organism. The answer is true because the isozymes will are basically different enzymes that are performing the same function. And if you delete all the enzymes that are performing that particular function of an essential reaction, catalyzing an essential reaction, if you delete all the enzymes associated with that, then the cell will not survive. So it will kill the organism, and the answer is true. Okay. So you're given this particular reaction R9, sorry, okay, which actually converts M8 to M9 and M10, and does not carry flux as the M8 metabolite is not produced when glucose is used as a substrate. When xylose is used as a substrate, R9 carries flux when basically M8 is produced. So R9 is a conditionally blocked reaction. So when there is glucose as a substrate, it's a blocked reaction. When there is xylose as a substrate, this reaction carries flux, so it's not a blocked reaction. So it's basically a conditionally blocked reaction. Depending on the available metabolite, it is blocked or not blocked. So it's a conditionally blocked reaction. Right, moving on. Okay, so you're given these two reactions, R7 and R8. So in R7, M8 and M6 gives you M7 and the gene A is involved. In R8, M8 and M9 is involved. Gene B is involved, giving you gene M10. So if there is no reaction producing M9, you are asked what is this? What is the like what are the two statements? Firstly, M9 is a dead end metabolite because M9 there is no reaction that is forming M9, but M8 is not a dead end metabolite because it is formed from gene A and gene B. So two different reactions are forming M8. So M9 is going to be a dead end metabolite, and yeah, that's the answer. You know. The answer to this is false. The statement was the biomass reaction will carry flux if at least half the number of metabolites that are part of the linear reaction are produced. That's not true. There is no relationship between the number of metabolites that are part of the linear reaction and like if it is half or like whatever the number is, the flux does not depend on that. The flux depends on what biomass is getting used up and what biomass is getting produced. That's all. So yeah, moving on. I think this is yeah. I think this is a better question. Okay, so you are given these five reactions. You are told ki in the growth medium, the three metabolites M2, M1, and M5 are present. Right. So you are asked to tell which of the statements are true. So if M1 and M2 are giving M3, M3, M1 giving M4, M2, M4 giving M9, M3, M5 giving M9, and M9 is the biomass, right? So R3 and R4, if you delete, so first option, R3 is a single lethal. So if you delete R3, there will be no production of M9, but that production will be taken up by R4, right? So it's not a single lethal. Now R3 and R4, if you delete, there will be no production of M9, which is the biomass. So these are double lethals, right? If you delete R1, which there is no production of M3, if there is no production of M3, no production of M4 or M9. So yeah, so if you delete R1, it's a single lethal, right? And R2 and R5, if you delete R2, you would lose M4, and R5, it's basically biomass only. But M4 can be uh, yeah. So R5 is a biomass reaction. So basically, it's just the M9 is getting converted to the biomass, but it's not a single, it's a double lethal because you can generate M9 without uh, R4, R1, R2 and R5. Sorry. Okay, so you are given these four reactions, R1, R2, R3, R4, and the genes that are associated with it. Now, if let's go one option by one option, right? So gene F is a single lethal. So if you remove gene F, you are removing this particular R4 reaction, and M6 is not getting produced, which is the required metabolite for cell survival, right? Yeah, and M1, M2 is in growth media, right? So M6 is not getting formed, so that is the cell will not survive. So gene F is a single lethal, that is fine. If you talk about gene A, so if gene A is not there, gene A and gene B is required for R1. So if gene A is not there, R1 uh, will not happen, and M3 and M4 will not be formed. So M5, M7, and M6 will not be formed. So it actually is a single lethal. Gene A is also a single lethal. Right? Gene C and gene E are double lethals. If you delete both C and E, uh, you're not forming M7, but this is not an issue because you can easily form M5 and M5 can form M6. So That is not an issue. M6 will be formed even if gene C and E are both deleted. So that is not the correct answer. Gene C, D, and E are triple lethal. So let's consider this. So C, D, and E. So R2 and R3 you're removing. You're producing M4, but you're not producing M5. So basically, M6 will not be formed, which is required for cell survival. So it's gene A, sorry, C, D, and E are triple lethal. So that is the correct option again. Let's move on to the last question. Okay, so in this question it says, "Okay, using the metabolic model of an organism, FACOF was carried out enforcing the flux through the reaction 5 at every step and noting the flux reaction change in reactions 1, 2, 3, and 4. So these are the four so these are the four reactions that were noted. Flux was noted for, and this was an observed distribution, right? Um, the following was observed for different fractions of maximum flux enforced, labeled from decreasing to increasing fractions, zero point 0.1, zero point 0.2, up to 0.5. So this is an increased, so this is a fraction of a product produced, and these are your basically flux at a particular reaction. So which reaction is our possible targets for overreaction? That is the question. The reaction 1, if you consider, it started out well, it increased, increased, and increased. But same is not the case for other reactions. So if you see the orange part, reaction 2 stayed the same across all four. The yellow bar or bar 3, it actually decreased across when the fraction of product increased, and reaction 4 actually had oscillating behavior. So reaction 1 is the ideal target for overexpression, because if you overexpress this, your final product, you'll have a very high overexpression. Okay, so that is all. Thank you for joining. Thank you for seeing, because nobody is currently here in the meeting, but yeah, thank you and see you next week.